Hello. So a lot of students have been struggling with the Catalase Enzyme Lab that didn't finish it before we left. So I'm going to create a video that kind of shows you the process of creating that Catalase Enzyme Lab and writing it effectively. So let me start. I'll share my screen with you. Screen is shared. And let's open some documents up. So to write the Catalase Enzyme Lab, you need your class data. So this is a data sheet from a group in period 3. This particular group looked at how enzyme concentration affects the rate of reaction. So when I look at their document, they have graphs for each trial that were used to calculate the reaction rate. In this particular one, I have 2.52 milliliters of oxygen produced per second as the rate of reaction. And then most importantly, I have this graph. And this is the graph that you want to be keying on more than any other graph because it has the catalase concentration versus the rate of reaction. So you're going to open up a document that you can eventually turn in. Let me title it Catalase Enzyme Lab. And let me put some things down that I'm eventually want to include in the lab. So I need a prediction. I need a data section. I need a data analysis. And I need an error analysis. The first thing I'm going to do is I am going to move those graphs. The reason being is I can't really grade your lab report without seeing the graphs, particularly the last one, because I have no idea what happened in your lab and how that relates to your prediction if I can't see your data. So let me move those. So to do that, I would go to the chart, copy chart. Then I like to go Control V to paste. And then I go to the next one. Let me move my copy chart. Go here. I'm not going to do this for all of them, but you would do that for all 6. But most importantly, I 100% need this graph right here, the most important graph of your independent variable catalase concentration versus the rate of reaction. So I'm going to copy that chart. And then bring it into my document. Great. Awesome. So this particular group was looking at enzyme concentration. So you can go to many websites to find what your prediction should be written off. Like I found this one website that looks at factors affecting enzyme activity. It talks about temperature, pH, which we did not do, concentration of the substrate, and the enzyme. For me, I am going to be focusing, of course, on this one, enzyme concentration versus the rate of reaction, and writing my prediction to describe this graph. So something along the lines of, as catalase enzyme concentration increases, the rate of hydrogen peroxide decompose will increase until a certain point when the rate of reaction will level off, or plateau is the kind of better word. But here I have a prediction that now I can actually respond to. So let me go down here and kind of show you something cool. So this was this group's graph. If I go back to kind of what this should look like, I think I can kind of draw it on here. Yeah, I can annotate this. I should have been getting something that kind of looked like this. So of course, this group has a bunch to talk about. They kind of have a sort of a plateau building here where these points level off, but this first part is mightily odd. Particularly their lowest concentration seems to have way too high of a reaction rate for what they actually got. So in my data analysis is where I would respond to my prediction. So I would talk about those things. I would talk about, whoa, this lowest concentration is way off. It should have been my lowest reaction rate, but it was actually my highest. That's not right. Well, I kind of got a plateau, but this point right here really doesn't fit within that plateau that I expected. I'm responding to my prediction using my lab data, particularly focused on this reaction rate versus catalase concentration graph. Then finally, let me get rid of my line. There we go. I do my error analysis where I talk about problems in the lab, particularly problems related to the trials that did not match my prediction. So we're looking for a paragraph here where you respond to your prediction, and then another paragraph here where you talk about the problems. If you get your prediction, your graphs in, particularly this last one, a paragraph where you respond to the prediction, and a paragraph talking about the problems, you will be good to go for this lab report. If you have more questions, let me know. I can certainly help. We can even do a Google Hangout where I can walk you through some of these steps. If you're really struggling, I'm absolutely willing to do that and provide you this support on the lab. We still have one more lab report to do this year, which you'll be using data that I actually collect in my basement. And knowing this process and reviewing it again will help you not only here, but also later to come. I hope this video helps, and let me kind of stop my sharing and then stop